Ever worried about drink tampering? It's definitely something people think about. So today's deep dive is into some really cutting-edge science fighting this exact threat. We're looking at the rapid detection of date rape drugs, DRDs in beverages. We're unpacking a pretty interesting 2024 review I found in Trends in Analytical Chemistry. It's called Current Trends in Rapid Electroanalytical Screening of Date Rape Drugs in Beverages. Long title. Our mission basically is to figure out how these electrochemical sensors are becoming, vital tools for checking drinks on the spot, making the invisible visible.
Yeah, and it's so important because, well, just assessing drug-facilitated crimes or DFCs is incredibly difficult.
Mm.
Often victims experience amnesia or there's fear involved. It's an insidious threat. There was one report really stark finding that 75% of rape victims had actually been drugged.
Yeah.
And these DRDs, we're talking about things like benzodiazepines, ketamine, GHB, they're often designed to be missed. Tasteless, odorless, colorless sedatives, almost impossible to spot without some kind of tool.
Right, so you've got this hidden threat, basically. And it's not just the drugs themselves that are the problem, is it? It's the drink, too. Like all the sugars, the alcohol, different flavors. That must make detection really hard, especially outside a lab.
Precisely.
Yeah.
That's the core challenge for on-site screening, all those potential interferences. But connecting this to the bigger picture, that's exactly where these electroanalytical tools really shine. They tend to be low cost, the setups are simpler, and many can even link up with a smartphone. We're genuinely moving towards a sort of plug-and-play situation for detection.
Plug and play. Okay, so let's get into some specifics there because this is where it gets really interesting, right? Starting with benzodiazepines, BDCs. You said they're the most studied.
Yes, in beverages they are. That includes things like flunitrazepam or Hypnol, which actually was pulled from the EU market specifically because it was used in DFCs.
Wow. But the tech is catching up. You mentioned wearables.
Exactly. Innovations like these wearable 3D printed e-finger devices, like a tiny sensor you could wear almost. Lets you check your own drink.
In anything, like beer or spirits.
Yeah, the research looked at whiskey, vodka, gin, beer. The idea is self-checking right there, right then, empowering people.
Okay, and not just BDCs. What about ketamine?
Ketamine KT is another big target. Similar progress there. We're seeing paper-based microfluidic devices and that e-finger tech again for detection in stuff like whiskey or orange juice.
What about newer threats, like those new psychoactive substances, MPS, or even fentanyl? Is research looking at those?
It's more limited, but yes, it's growing. It has to. There are concepts like a lab-on-a-glove sensor tested for fentanyl, which shows promise. You can potentially adapt that kind of tech for beverages. Someone even found a way to detect morphine in coffee and milk, interestingly.
Okay, but let's talk about the one that sounds like a real headache. GHB, gamma-hydroxybutyric acid.
Right.
You mentioned it earlier. Why is GHB such a particular challenge for these kinds of sensors?
Oh, GHB, yes. It's tough for several reasons. It's colorless, odorless, mixes easily in water, and critically, the body gets rid of it very quickly. Often, like only 1 to 5% might be recoverable in urine just hours later. Makes forensic work hard. But for these sensors, the main issue is that GHB is non-redox active.
Non-redox active, meaning?
Meaning, it doesn't easily participate in the electron transfer reactions that most electrochemical sensors rely on.
Yeah.
So under normal conditions, these sensors just don't see it very well.
Yeah.
Or at all. It's a huge research gap. Seriously. Only one report I saw managed to detect it in alcoholic drinks, and they did it indirectly by seeing how GHB suppressed the signal from ethanol oxidation.
So like detecting its shadow almost?
Kind of, yeah. It shows we need really novel thinking, completely new platforms for GHB detection. The need is immense.
Okay, so GHB is the big hurdle, but the overall goal remains that plug-and-play idea, making this tech truly usable for anyone, anywhere.
Absolutely.
Like you could maybe scan your drink's electrochemical fingerprint, and then if you're suspicious later, scan it again and see if that fingerprint has changed, instantly revealing if something was added.
That's exactly the vision. Compare the before and after. It reflects this critical push for tools that are cheap, portable, disposable, but also highly precise and reliable. Moving detection out of the lab and into the real world, enabling prevention right where it counts.
So reflecting on all this, what really stands out to you from this deep dive? The science is clearly moving fast.
For me, it's the sheer ingenuity being applied to tackle these invisible threats, especially when facing fundamental challenges like with GHB. It really highlights how science responds to critical real-world safety problems. The next big step, I think, is bridging that gap from lab innovation to truly accessible, everyday tools that could really change things.